Hello and welcome everyone to Bio-Techne's COVID-19 virtual symposium. My name is Yas Haidari and I'm the Protein and Cell Biology Product Manager for Bio-Techne Europe, and I'll be your host for today. Firstly, I'd like to thank everyone for attending from wherever you are joining us from the world today. We have a staggering 600 plus people joining us today from across the globe. Thank you everyone, it's great to see. We have an excellent lineup of speakers for you today, which will hopefully make for great sessions. You will also find an abstract booklet for this event in the handout section on the right side of your screen. So this booklet has the program and other information. Also, to keep proceedings lively and fun, and to help with everyone's COVID-19 research at this time, we are offering every registrant a chance to win a golden ticket worth $1,000 on reagents for COVID-19 research. So to win, you just have to answer the survey towards the end of the abstract booklet. You just click here. It takes 1 to 2 minutes max, and the odds should be good as people don't like filling forms. So whenever you get 2 minutes from now until 2 weeks time, the deadline shows there. On the subject of interactivity, throughout today, we invite you to submit any questions you have for our presenters using Ask a Question box on the GoToWebinar control panel, again on the right side of your screen. We encourage you to indicate if you would like the panel or a specific presenter to answer, so we know who to address your question to. We will have a question and answer panel with our speakers right at the end of our session. This is so we respect everyone's time, finish the symposium according to the schedule. In case there are any questions left unanswered by the end, we will email the answers, so no worries on that count. Yeah, so it's really our aim to make the symposium as interactive as we can make it. We want to hear from everyone. So we will be using a software called Slido throughout today's event. So if you would kindly head over to the website www.slido.com, you will be able to sign in with today's event code, and that's COVID-SYMPO. We'll just give everyone a minute to do that. Yeah, so once you've gone to that website, and if you can open the Slido and keep it open, this is the gateway which we can ask questions and you can express your opinion, and we can hear from you. So Slido question 1. If everyone's ready, where are you joining us from today? Where are you joining us from today? Oh wow, we're getting people joining from everywhere. So I see UK, Malaysia, San Diego, Qatar. Someone just put home office. Indonesia, Boston, Chile. Wow, Canada, Maryland, Minneapolis, Malaysia, New York. We have some people from New York, Texas, Brazil. Wow, we have people from Brazil. We give everyone a little bit more time because this is the first question. I think I saw Japan a little bit earlier. Ireland, excellent. Okay, so we seem to have a lot of people from UK, Qatar, France, Maryland, Boston, and India. So everyone, welcome. It's great to see everyone from all different corners of the world. And so thank you for participating in this first question as well. We will have other questions coming up, so please keep that page open. So next, we go to our program just to give you an overview. Our agenda for today will evolve around our presentations, interspersed with interactive questions that you just participated in, and we'll also have some live polls, and we will finish with a question and answer panel right at the end. So our program will kick off with a talk by Anthony Pearson, our very own Director of Protein Business. He will give us an update on the latest on coronavirus proteins and relating reagents from Bio-Techne. Then we will hear from Joep Bomer, joining us from Utrecht in the Netherlands. Joep is from Hans Clever's lab, and he will tell us about how he's utilized intestinal organoids to investigate SARS-CoV viral entry mechanism, the work that he's recently published in Science, as well as a little on ongoing studies. We'll complete the presentations with a talk by our scientist, Jamie Van Etten, and the talk will be focused on profiling innate and adapted immune cells in COVID-19, exploring in vitro approaches to mimic cytokine release syndrome and its subsequent impact on cells of the immune system. So now it's time to introduce our very first speaker, Dr. Anthony Pearson, joining us from Minneapolis, Minnesota. Anthony is our Senior Director of the Protein Business at Bio-Techne. He was trained as an embryologist, where he focused on elucidating the molecular mechanisms of Wnt biology in the context of cardiac development and human genetic diseases. Anthony worked in the stem cell field for multiple years, developing methods for reprogramming somatic cells to pluripotency and transdifferentiation of cells from one type into another using mRNA overexpression. Due to the recent COVID-19 pandemic, Anthony's protein unit has prioritized the development of COVID-19 relating protein reagents, which he'll be telling us more about today. Good morning, Anthony.
Good morning, Yaz. Thank you for the introduction. Yes, as Yaz said, I am Anthony Pearson. I am the Senior Director in charge of the Protein Business Unit at R&D Systems, part of the larger Bio-Techne Corporation. And I'm excited to talk about our new coronavirus protein reagents that we're developing at Bio-Techne. Without further ado, I'd like to get into the presentation. First, a little quick brief history about R&D Systems and Bio-Techne. It all started in 1976 when R&D Systems was founded in Minneapolis, Minnesota, United States. As you can see on the bottom right corner, this is a picture of beautiful Minneapolis from the Mississippi River. Above is the corporate headquarters in Minneapolis of R&D Systems, Bio-Techne. We have a long, rich history of making recombinant proteins, with our first protein, TGF-beta, purified from platelets back in 1985, was released to market. The first recombinant protein was IL-2, released to market in 1988. We have over 5,000 protein products in our catalog and 51 GMP proteins. R&D Systems has a rich history of selling not only proteins, but also antibodies and ELISA kits. With the acquisition of Tocris back in 2011, we also offer small molecules and other biologics as well to our customers. We have over 2,600 employees worldwide. And in 2014, after R&D Systems was acquiring multiple companies over the years, the decision was made to rebrand the corporation to the Bio-Techne name, of which R&D Systems and all these other companies are now part of the larger Bio-Techne Corporation. So now I'd like to get into coronavirus biology and some of the proteins that we've been working on. The proteins I'm going to discuss today in detail would be spike proteins on SARS-CoV-2, as well as MERS and the original SARS virus. These spike proteins are trimeric proteins on the surface of the viruses. They, for SARS-CoV and SARS-CoV-2, they bind to angiotensin converting enzyme 2. For MERS, they bind to a different receptor called DPP4. And there's a great demand for spike proteins for several reasons. Number one, antibodies against the spike proteins are very sought after in the pharmaceutical industry as therapeutic reagents. In addition, spike proteins can be used for serological diagnostic testing to try to find antibodies in patients that have been infected with COVID-2. We're also going to talk a little bit about our nucleocapsid proteins These are viral binding proteins within the virus. And the reason that these are important as biological reagents is they're shed in high amounts in the serum and nasopharyngeal aspirate during the first week of an infection. And nucleocapsid proteins have been used quite extensively in antigen-down ELISA diagnostic tests as well as lateral flow testing. And finally, I'd like to talk about the proteases that we've isolated and purified that we sell on our website that many pharmaceutical companies are interested in drugging. The papain-like proteases and the C3L proteases from MERS, SARS-CoV, and SARS-CoV-2. To start with the nucleocapsid proteins, we already have 2 of these on our web available for you to purchase. We have the MERS-CoV-2 nucleocapsid protein full length with a C-terminal His, as well as the SARS-CoV-2 nucleocapsid, again, full length with a C-terminal His. We found that the SF21 insect system using our baculoviral delivery platform has worked very well to make these nucleocapsid proteins. We are also working to bring the SARS-CoV nucleocapsid to market in short order as well. These are highly pure reagents, and we've done some internal testing with these, and they're very useful for serological ELISA assay development, lateral flow ELISA assay development, Luminex assay development, as well as immunogens for antibody development. So these are already on our website, and if you're interested, you can contact me with more information, but our product inserts have extensive information and specifications on them. Now I want to talk a little bit more about some really interesting biology, which is the spike proteins. Spike proteins are fascinating proteins. They're on the surface of the SARS-CoV-2 virus, as well as SARS, the original SARS and MERS. And for SARS-CoV-2 and for the original SARS virus, these trimeric spike proteins bind to ACE2 on the surface of our epithelial cells. This leads to a cleavage event by TMPRSS2 that cleaves the S1 portion of the protein away, leading to S2 portion of the protein binding to the cell surface and fusion with the cell and infection of the cell. And what we're going to talk about in the presentation today is the proteins we're offering for spike proteins. We've purified S1 domain portion of the spike protein, the full-length spike protein encompassing S1 and S2. Of course, not with the transmembrane domain because we purify from the conditioned media of our cells. We've also made receptor binding domain versions of the spike proteins that have been shown to bind to and articulate with the ACE2 protein. And we'll talk more about the details around these spike proteins in subsequent slides. Here are the spike proteins that we are working on or have in inventory already. The receptor binding domain SARS-CoV-2 proteins we've made with either FC fusions or His C-terminal fusions, depending on what our customers may want. We've made these proteins in HEC, CHO, insect TN5 cells, and we're providing many different flavors to our customers because, as you'll see later in the presentation, the glycosylation patterns from these different RBD, His, and FC proteins are variable depending on the host used to produce them. We also have various versions of the S1 domain SARS-CoV-2 proteins in our inventory right now, and we make those in SF21 cells, in HEC cells with His or FC tags. We even have generated an S1 domain SARS-CoV-2 protein with engineered trimerization domains from the S2 domain to make a trimeric version of S1 protein if that's of interest to the customer. The S2 domain for SARS-CoV-2 proteins are still in process. We're purifying them this week, and these should be on our catalog in the next month. And finally, we have the full-length spike proteins made in either HEC or CHO cells with arginine mutations to prevent the cleavage events, as well as proline mutations to put these into the pre-fusion conformation, which is more sought after by our customers. Now at R&D Systems, we really go out of our way to show biological activity of our proteins. I believe it's something that differentiates us from the competition. And what you see before you here is ELISA data using our SARS-CoV-2 RBD-FC protein binding to ACE2 His protein. And on the right, you see SARS-CoV-2 RBD His protein binding to ACE2 His. And the way we've done this assay is we've coated the RBD proteins on the plate at 200 nanograms per mil. We're titrating in human ACE2 His as the ligand, and then we're detecting with a goat human ACE2 biotinylated antibody. And the ED50 for binding for each of these reactions is in the low nanogram per mil range, which is expected based on the literature. So they are tight binders to the ACE2 protein in ELISA workflows. Now, we also have a BioAccord T200 at R&D Systems, and we use this extensively to get true dissociation constants. And we've done a similar experiment where we've looked at binding of RBD-FC from SARS-CoV-2 with ACE2, or the RBD His version with ACE2. And again, we see low nanomolar dissociation constants for both the FC fusion and the RBD His versions of the SARS-CoV-2 spike proteins. So tight binding as would be expected based on the literature. Now we've also gone on to make, as I stated earlier, SARS-CoV-2 S1 fragments, which are from 1 to 681 amino acids with a His tag on the C terminus. A similar workflow where we've coated the SARS-CoV-2 S1 protein on the plate, titrated an ACE2 His, detected with a goat anti-ACE2 biotinylated antibody, and we get low nanogram per mil ED50 levels for this as well. And here on the right, we have the full length spike protein with arginine mutations, as well as the pre-fusion conformation with the C-terminal His. And we see nice nanogram per mil binding for the longer spike protein to the ACE2 receptor in ELISA workflows. Again, surface plasmon resonance to look at true dissociation constants were done. And again, both the SARS-CoV-2 S1 His binds to ACE2 His with low nanomolar dissociation constants as expected. And the spike protein, which encompasses the entire spike minus the transmembrane domain with the C-terminal His, has even a sub-nanomolar dissociation constant in SPR experimentation. So we're very happy with the bioassay results for these proteins, and we either have these on our website already or they will be within the next couple weeks. Now, we looked extensively at the spike protein. This is the full-length spike protein, and ours is a beautiful trimer, which is desired. We made this spike protein for SARS-CoV-2 with, again, the arginine mutations and the pre-fusion conformation for better exposure of the RBD portion of the molecule when you're trying to make antibodies. And the predicted molecular weight of the monomer is 134 kilodaltons. But what we see is a nice trimeric form for the CHO stable line derived full-length spike protein, as well as the His transient derived full-length spike protein. So we're very happy with this. And we've looked at competitors' spike proteins, and we don't see these same species. We see more oligomerization with competitors' material. We have multiple ACE proteins already on our website. As the one we've been selling for a while is ACE2 with the His tag on the C terminus. We're also just about to bring to market an FC fusion of ACE2 human. We're working on biotinylated versions of ACE2, AVI-tag versions of ACE2 as well, which have a single biotin per molecule. We sell mouse and rat ACE2 proteins already, and we also are working on a porcine ACE2. In addition to the ACE2 receptors, which I are showing you again as products on this, we also have other coronavirus receptors if you're interested in these, like DPP4, which is the MERS receptor. We have the aminopeptidase N, which is a receptor for other coronaviruses listed here. And then we have some accessory coronavirus receptors that have been implicated in this biology, like CA-CAM1, LY6E, and Emphrin, if that's of interest to our customers as well. So you might ask, what are customers doing with these reagents? And I think that a major portion of what our customers are doing is they're looking for blocking antibodies, either against ACE2 or more importantly, against the spike protein on the surface of the SARS-CoV-2 virus. As we know, a vaccine is in development by many different pharmaceutical companies, but the time frame of when that will happen and the ultimate efficacy of the vaccines is completely unknown. Another issue we'll deal with is that there's not always 100% compliance with taking vaccines, as we well know, and so many people won't take the vaccine. So we believe strongly that patients in the clinic that are sick will be treated ultimately with blocking antibodies against the spike or ACE2 in the future. And so what our customers are doing is they're binding the ACE, they're taking the ACE2 protein and the spike proteins and looking for blocking antibodies in ELISA workflows, or they're using an approach like we're using in-house as well, where they transfect into cells a full-length transmembrane ACE2, and then they can add our recombinant proteins as well as detection reagents in a flow cytometry workflow to look for blocking antibodies against ACE2 or spike proteins. We have done that internally at R&D Systems, and we have already identified multiple blocking antibodies. We have multiple ACE2 blocking antibodies that are already on our catalog that are well published already in high-profile journals. And then we have another reagent, Lamabody actually, that binds to and blocks the spike binding to ACE2 that has been validated in our flow cytometry assays. And then we have a whole bunch of other antibodies, monoclonal antibodies that we found in our screening that beautifully block the interaction. So they're not online yet as web reagents, but please contact me and I can get you in contact with the antibody business unit and they can work with you on sourcing these reagents. Now I want to move on to a fun project that we've been developing at R&D Systems, looking at the glycosylation patterns of these spike proteins. As many of you likely know, these spike proteins are highly glycosylated. And we have been developing reagents to probe and footprint O and N glycosylations on recombinant proteins. When we initially set off to make the RBD His protein, which we believe is the most desired protein in the mix for SARS-CoV-2 for many reasons, we used a shotgun approach where we used our CHO, HEC, SF21, as well as TN5 insect host systems to make the RBD His protein. And we are curious in looking at the glycosylation footprints from these various RBD His proteins for SARS-CoV-2 using our novel footprinting analysis. And what we do at R&D Systems is we have over 800 different enzymes that we already sell, and many of these are focused on glycobiology. Glycosyl transferases like ST3-gal1 are highly specific enzymes that recognize specific structures on O-glycans and can add sialic acid to galactose on those side chains. And we also have made fluorescently labeled sugars like sialic acid and fucose to use these enzymes to add these sugars to glycosylation chains on our proteins in a footprinting workflow. And so what we have here is, if you look on the right, ST3-gal1 plus Cy5 sialic acid will add a sialic acid to a galactose on the chain of O-glycans. Neuraminidase removes sialic acid. So neuraminidase will remove sialic acid, something like this. And then endo-FEF, also called O-glycosidase, recognizes core1 type O-glycans and will remove them from your protein. So we've taken our proteins made from the 4 different host cells, and we've done 3 reactions. The minus reaction is just adding ST3-gal1 enzyme with Cy5 labeled sialic acid. And this will basically add sialic acid, fluorescent sialic acid moiety to any galactose on these chains. Neuraminidase will strip off the sialic acid. And then if you do a reaction after neuraminidase, adding ST3-gal1 plus Cy5 sialic acid, it'll add the sialic acid back. This is a way to probe for, is my protein terminally sialated on O-glycans? And finally, we have the EF reaction, which uses O-glycosidase, which will recognize this core 1 type O-glycan. It will cleave it off. And then if you try to add sialic acid back, you won't see any addition of the sialic acid because you've removed the entire chain. And let's start here. If we look at the EF lanes below, which we've done just that, for CHO, SF21, and TN5, we see no bands there. What that means is that when you treat with O-glycosidase, you have recognized and removed the core 1 type O-glycan on these proteins. This data suggests that protein made from CHO, SF21, and TN5 insect cells contain core 1 type O-glycans on their surface. Now, if you contrast that with HEC-derived RBD His protein, you see a robust band there. That suggests that indeed HEC-derived RBD His protein does not have this modification, but rather has a more complex O-glycan on its surface, the core 2 type O-glycan. Clear differences if you look at the band in EF lane here compared to the CHO and the SF21. Something else to note is that when we do the neuraminidase treatment and then add back Cy5 sialic acid, you see a clear increase in signal from this lane to this lane. That means that the HEC293 cells are not only of the core 2 type O-glycan, but they also are terminally sialated, which is clear by the gain of signal from this lane to this lane. So I will have a summary slide to go over the O-glycan species, but the take home from this is that CHO, SF21, and TN5 have this O-glycan species, whereas HEC-derived RBD His has the more complex core 2 type O-glycan species that's terminally sialated. Now we've developed similar footprinting tools to look at N-glycosylation. And what we have here are new different glycosyl transferases and reagents that can add, like for example, ST6-Gal1 will recognize complex biantennary N-glycans and add a sialic acid to the end galactose on the chain. FUT8 is a different enzyme that recognizes the more immature N-glycan, high mannose N-glycans, and will add the fucose to the proximal position on the chain here. And so what we've done here is we've done similar reactions, but this is more complex, and we're adding 2 different enzymes to label either complex N-glycans or high mannose N-glycans. And to make a long story short, if you see red labeling on the gel here, that suggests that the protein has a complex N-glycan biantennary like this. And if you see green labeling on the gel, that shows that the insect-derived RBD His proteins have the high mannose N-glycan structures. And you can only see a little bit of green labeling over in the CHO-derived RBD His. So the summary is complex N-glycans are found on CHO HEC-expressed RBD His proteins, but not on insect cell-expressed proteins. And high mannose N-glycans are largely found on insect-expressed proteins and are a smaller percentage of the CHO HEC-expressed proteins. Again, CHO and HEC have complex biantennary N-glycans. The SF21 and TN5 that have the green label have more of the immature high mannose N-glycans on their surface. Here is a summary of what we find from our glycan footprinting analysis. RBD His protein made in CHO cells have complex N-glycans that are not terminally sialated, and they have core 1 O-glycans. RBD His made in HEC293 cells have complex N-glycans that are terminally sialated, and they have sialated core 2 O-glycans. And RBD His made in insect or TN5 cells, both insect cells, have the more immature high mannose N-glycan structure as well as core 1 O-glycans. And you might ask, well, what does this do to the binding affinity of these proteins to ACE2? We've tested that in ELISA, and we really don't see a huge difference in the binding affinity, both in ELISA or SPR experimentation. But some of our customers may want these different glycosylated versions of RBD His proteins to inject animals to make antibodies, or for serological workflows to test patients' blood to see if they have antibodies against the spike protein. So we're giving our customers different versions of certain proteins with different glyco footprints, depending on the needs of our customers. We have all these enzymes already on our catalog, and we're working hard to bring the fluorescent labeled sugars on our catalog very soon. We have an application note that will be on our website in the next 2 to 3 weeks to describe the methods for this footprinting of our proteins with our enzymes and fluorescent sugars. So look out for it. It's a really exciting new technology we're developing, and it allows us to look at glycosylation patterns without mass spec. Finally, I want to talk about some obvious drug candidates in the genome of the SARS-CoV-2, which are the proteases. Whenever you think of druggable candidates for viruses, proteases are generally the first thing that comes to a person's mind. And the SARS-CoV-2 virus is similar to other viruses. It makes a large viral polyprotein that is subsequently cut with two proteases, the papain-like protease and the C3L protease, that cuts this large polyprotein into its functional units, the different proteins of the virus. And if you can disrupt these proteases with small molecule drugs, you may have a very nice therapeutic on your hands to fight this COVID-19 virus. And we have already made the papain-like proteases for SARS-CoV-2, MERS, as well as the original SARS virus. We've just released the SARS-CoV-2 3CL protease, and we're working on MERS and SARS, the original SARS version as well, that will be coming on our website within the next month. Here's a look at the 3CL protease from SARS-CoV-2. And it's a highly pure protein if you look on a page gel with Kamasi blue staining. Our mass spec data shows that it's the predicted molecular weight. We've developed bioactivity assays. All this work for the proteases is done in Boston at Boston Biochem, an affiliate of ours that's within the protein business unit. And they do very nice enzymatic work. And what they've developed here is some bioactivity assays to look at the activity of the 3CL proteins. And they've developed a novel peptide rhodamine reagent that, when cleaved, fluoresces. And this is a nice bioactivity that you can test for the activity of SARS-CoV-2 3CL, the original SARS 3CL enzyme, and of course we have a negative control. This particular peptide was specifically designed based on the recognition sequence of the SARS-CoV-2 3CL enzyme, but it also works for the SARS 3CL. And we're working on a new peptide reagent that's more specific for the SARS 3CL right now. But they're nice bioactivity assays that are amenable to large-scale small molecule drug screening, if that's interesting to you. And of course, we can provide these enzymes in bulk. The papain-like proteases, similar story. We've isolated these and purified them. They're highly pure. And we've developed bioactivity assays. In this case, these papain proteases are known to cut ubiquitin as well as ISG15. In this case, we're using ubiquitin rhodamine in a similar fashion to the previous slide. And these enzymes are beautifully active. And again, these activity assays, we can sell you the proteins, we can sell you the reagents, and help you with figuring out how to run these assays to run your small molecule candidates on these enzymes to see if you can find blockers. So this is my final slide. I'll leave you with this. Why choose Bio-Techne for your coronavirus protein needs? The number one thing that I generally tell customers is quality. We go out of our way to provide our customers a lot-to-lot consistency. They're highly pure proteins. Endotoxin is very low, aggressively low. We do N-terminal identity testing on all of our proteins to make sure you're getting the right protein. The bioassay activity testing has to be within 2-fold of reference slots. and we have over 1,000 bioassays for our proteins. I used to be a manager in the bioassay department. I believe that our bioassay capabilities are second to none, and this differentiates us from the competition. When you get a protein, it's going to be the same as the last lot of protein you got. Analytical testing, we have our impressive analytical suite. I showed you some SEC data. We have multiple mass specs. We do all sorts of analytics on our proteins, and we really like to understand what's in the tube for our customers and make sure if it needs to be a dimer, it's a dimer. If it's supposed to be a trimer, it's indeed a trimer, and not some oligomerized aggregated material. We have our quality standards, ISO 9001 and ISO 13485. We work with customers on bulk orders, custom bottlings. We have our industry-leading customer service, technical service, and sales departments. Please contact me if you're looking for a protein that may not be on our catalog. For every protein we sell, we have multiple different flavors of the protein with different tags, different amino acids, different hosts. So please reach out to me, and we routinely send out non-catalog proteins to interested customers. We have a rich history of recombinant protein experience over 35 years. We pride ourselves in making the most challenging proteins, WNTs, BMPs, TGF-betas, noggin, dimeric integrins, IL-12 family members, and like I showed you today, the coronavirus spike protein that's a true trimer. We have rapid transient production capabilities, scalable, stable production capabilities, and we work with a lot of major pharmaceutical companies on custom projects. So remember, time is money. Do you have time to do it right the second time? Please come to Bio-Techne for your coronavirus proteins and reagents. Thank you very much.
Thank you very much, Anthony, for that great presentation. So everyone, just to remind you all that questions, there's a question and answer session at the end. Please continue to write your questions in the box. And just thank everyone in advance. And also, Anthony, I'd like to take this opportunity to thank the lab for all their hard work making these proteins and releasing these reagents. Not easy under lockdown, so please convey.
Yeah, we will. We've been working 24/7 on these proteins, and it's the energy in the lab has been kind of fun to watch. And it's a very difficult disease plaguing all of the world, but it's also an interesting challenge to tackle. And I believe that the scientists and staff in the protein business unit are really stepping up to the challenge. So it's an exciting time to get these into our customers' hands for sure.
Yeah, I'm not sure how widely known it is, the complexity in the glycosylation pattern of the proteins. I was quite fascinated.
Mm-hmm.
Okay, so now it's time for some more interactive questions. So question number 2: Are you still able to work on experiments in your laboratory? Are you working in a shift system? Are you still under lockdown? Okay, so there's some lab action happening out there. Okay, so that's promising. So it looks good. Globally, maybe we're starting to get rid of lockdown, 41%. It's shifting a lot. Okay, it looks stable. Okay. All right, so the next question, please. Yeah, so for those of you who have started or are starting, this is a fun question. How are you feeling about restarting lab work? Yeah, so the pipetting skills, they might be a bit rusty. It's been a few weeks. Okay, excited. Yeah. Yeah. I remember like there was a gap when I was writing my PhD and I wasn't doing any lab work. I went back and I was very slow at pipetting everything, so I had to leave more time to do anything. Okay, so we're getting Most people seem excited, which is great. Enthusiastic, so it's good. All right. I think so. It's good to see a positive, excited response going back to lab work. So thank you everyone for your participation. We'll have some more interactive sessions. Our next speaker is Joep Bomer, now joining us from the Hans Clevers lab, Utrecht, the Netherlands. Joep is currently undertaking his PhD studies where he's been using organoid models to study the signals guiding the homeostatic turnover and generation of differentiation lineages in mainly mammalian tissues. Lately, Joep has been using the intestinal organoid system in order to investigate SARS-CoV-2 viral entry mechanism and the subsequent epithelial response to coronavirus. Hello, Joep, and welcome.
Thank you, Yes. Thank you for the kind introduction, and good morning, afternoon, evening to everyone around the world. I'm Joep Bomer, a PhD student from the Clevers lab. We use organoids, to try to use organoids to model coronavirus infections. Just want to state that we are not virologists, so we don't know much about viruses, so please be gentle with your questions. So a brief introduction to our systems, adult stem cell-derived organoids. So every organ in an adult has a stem cell population that replaces cells that are lost during normal wearing and tearing. And these adult stem cells in each tissue can only make the cells of that particular organ. In the lab, we have developed methods to isolate those individual stem cells, these adult stem cells, and derive 3D cultures, resembling the tissue. So these organoids, they can expand indefinitely. They are genetically stable, 3D structures, and importantly, they have the different cell types of the corresponding tissue only. So not like IPS, induced pluripotent stem cell-derived organoids, you can derive those into any kind of body tissues. From adult stem cells, you can only make the tissue that it's coming from. And another important feature is that we really only grow the epithelial side. So adult stem cell-derived organoids have been established from almost every adult tissue, epithelial tissue, different variations on the growth media. That basically the growth medium is the replacement of the mesenchyme, because you don't have that in the culture, so you need to mimic the signals from the mesenchyme. And as well, the organoids are embedded in a three-dimensional matrix that mimics the extracellular matrix. For that, we use the Cultrex BME. But there are many variations to that. And recently we were before focusing on mammals, but we also stepped down the evolutionary tree and have done this for the first time for reptiles. And we think this is maybe possible for all animals. So in our lab, we have before used organoids as a model of viral infection. So this is an example work by Norman Sachs in our lab. A German postdoc who worked on RSV. It's a very deadly lung virus, killing more than 100,000 kids, children around the world every year. Not so much known about that virus, and he used lung organoids as a model to infect them. So he could get very good affection in many human lung organoid lines, looked at with RNA sequencing at the epithelial response. You saw the upregulation of many cytokines, which are known to attract neutrophils. And in a co-culture assay of these organoids with neutrophils, you could also show infiltration of neutrophils into the organoids. So in that sense, a very good model, because this is something that's also seen in vivo. Some extraordinary phenotypes he saw with live cell imaging is that these organoids, upon infection, start moving like crazy. Here in red, you see the RSV virus. The organoids become very motile and aggressive, you can almost say. They start fusing with other organoids. And this is something known from actual infected patients. Cells that are infected with RSV can make syncytia, basically fuse with other cells it's a means for the virus to spread by fusing with other cells. So in that sense, this organoid really mimics this phenotype as well. Another example is the norovirus, which is actually an enteric virus. very well known from, or notorious from cruise ships maybe, nasty virus. But until 4 years ago, there was no way to grow this virus in an in vitro culture system. And the science paper changed that. They used, this study used intestinal organoids, and they could basically amplify almost any norovirus strain. They showed that in the absorptive enterocytes, here marked by villain, they could get replication the full life cycle of the virus. And they also noted that some strains that exist in human populations couldn't replicate, but dependent on the addition of bile. Bile is a very common component of the intestinal lumen, and these viruses really dependent on bile. And still don't know exactly why, but it's an interesting feature of the virus you could not have captured easily in vivo. So at the beginning of the COVID-19 pandemic, we were interested in using the organoid model, the adult stem cell derived organoid model, to study coronavirus infections in general, particularly SARS-CoV-2. And we realized at some point there were some initial reports in February of symptoms in the gut of patients with COVID-19. So diarrhea, about 1/3 of the patients, and also viral RNA in the stool of patients was very common. People even use it now as a measure of viral spread, measuring the RNA in sewers. It's a very good prediction of how much the virus has spread in a population. And also the ACE2 receptor, already introduced by Anthony, is ubiquitously expressed or almost throughout the whole epithelium in the intestine here, particularly high in the villus. And this is the entry receptor of the virus, so indicating that these cells could maybe facilitate infection. Because the intestinal organoid is our most mature model, we thought, hey, maybe we should start with using this as a potential way to model the infection in vitro. From ongoing studies, we already knew that our organoids could as well express ACE2, particularly the enterocytes. This is single cell sequencing, showing the expression levels in single cells. These blue and green, yellow bars are enterocytes, very high expression of ACE2. In gray, stem cells, barely any ACE2, is consistent with the expression data from in vivo. We also noted that activation of BMP signaling, a particular signaling pathway, greatly increased the expression of ACE2. So when we knew that, that was actually a reason for us to team up with virologists to use these BMP activated cultures in particular as a model for corona infections, SARS-CoV-2 infections in organoids. So we teamed up with the Erasmus Medical Center in Rotterdam, coronavirus experts there, Bart Haagmans and Marianne Koopmans, and we, together with them, looked at organoids differentiated towards different cell types, and then infected them with either SARS-CoV, the original virus from 20 years ago, or the new SARS-CoV-2. And we differentiated the organoids towards enterocytes, and then both in the presence and absence of BMP, we made secretory cells, which is the other major differentiated cell type in the intestine. And we also had a culture where cells were still dividing, resembling stem cells, and these dividing cells have very little ACE2. This is work from Mart Lamers. He quantified the replication of the virus in the organoids. He both used viral RNA measurements for that, as well as a life titer determination in the supernatant of the cultures. And to our surprise, the organoids were an expansion medium. So these are the ones that have mostly stem cells with very low ACE2 expression. Actually was the one to host the replication of the virus most efficiently. So indicated here in these graphs. We could also get replication in differentiated organoids. We didn't see any difference with the addition of BMP. And also the secretory cells didn't really host any more efficiently the life cycle of the virus. Here, RNA expression data, again confirming that this ACE2 is really 1,000-fold higher in this BMP condition than in the stem cell condition. But it seems that maybe low ACE2 level expression levels are enough to facilitate infection. So this RNA is also consistent to what we see on protein. Actually, this is staining with the R&D ACE2 antibody. We see in the expansion conditions barely any ACE2 on the membrane, and not really visibly, sometimes a little bit. And in the differentiated organoids that don't have much more efficient, basically not more efficient replication of the virus, there's way more ACE2, particularly high on the membrane, where you expect it. On average, very shortly after infection, 1 day after infection, we typically only see 1 cell infected per organoid. And over the course of the next 2 days, this infection spreads rapidly, on average 10 to 20 times more per organoid into that whole organoid. So there's really replication of the virus and spreading to other cells. Looking at what kind of cell types got infected, here in red, ApoA1, a marker of enterocytes, we could see indeed enterocytes being affected in the differentiated cultures. These are the nutrient absorbing cells of the intestine that have the highest ACE2 expression. So those are the likely suspects. But as I said before, proliferating cells, the proliferating cultures also hosted the infection very efficiently. And then here in KS67, marked by KS67, a proliferation marker, we could indeed see proliferating cells being infected. This is quantified. The percentage of organoids being infected initially is about 10%. Doesn't change so much over the course of 2 days. But the number of cells infected per organoid, so if there's 1 cell in an organoid that is infected, you get over the course of that 2 days a rapid spreading of the virus from cell to cell. So this is transmission EM performed by our collaborators in Maastricht, Peter Peters' lab. They could visualize the whole life cycle of the virus in these organoids and also noted apical shedding, particularly of the SARS-CoV-2 from the cells. Then we employed RNA sequencing to look at the epithelial response, so what genes are changing upon infection. And we particularly noted the huge upregulation of interferon-stimulated genes, what you would suspect the standard antiviral response. And if you look at GO-term enrichment analyses, you see typical expected things such as defense response to virus and things like that. We don't see upregulations of interferons type 1, like interferon alpha or beta or gamma. We only see type 3 interferons, particularly upregulated, such as IFNL1. And this is actual ELISA data. So this happens both on the RNA and protein level. These are very much upregulated. Our differentiated organoids, the data I just showed was from infected expanding organoids. The differentiated organoids, well, first of all, they differentiate really well. They upregulate all the expected markers of the different lineages that exist in the intestine. So these are enterocyte markers, and these are other cell types. They all increase upon differentiation compared to expansion. And including those, there's ACE2, greatly upregulated. But as I said before, not much more infection. And important to note here, there have been some reports that ACE2 can either be upregulated or downregulated after the infection by the virus. We don't really see any changes in this system in infected or non-infected cultures in terms of the expression levels of ACE2 or any other marker that we associate with the infection, such as the proteases. The differentiated organoids, looking at the RNA sequencing pattern of the infected organoids, we see the same genes being upregulated as the expanding organoids do upon infection. So that's also not different. We see the interferon-stimulated genes. We see type 3 interferons. So that suggests that the cell type that is infected doesn't really dictate what kind of response there is, at least in the intestine. And also these similar genes that we see upregulated in the intestine have been reported later in the lungs as well. So it might not be so much cell dependent what the response is. So we've been trying to look in these data sets for some surprising genes that would be potentially be druggable, or genes that are just not reported in relation to viruses or coronaviruses. And just to mention one example of one gene which was particularly striking in the intestine is ARC. ARC is actually was a not so well described gene until 2 years ago, when it was found that ARC in the neurons is a gene that is actually used to ship RNA between mRNA between pre-synapses and post-synapses. And what people think is that ARC is a repurposed GAG protein. GAG is the envelope protein of retroviruses. And this gene is basically got introduced in our genome and is now used to ship RNA between cells in a same way as viral capsids do that. So this ARC can form capsids just like viral capsid proteins do that. And we got this very striking upregulation of an infection. So maybe this is something abused by the SARS-CoV-2 as a mechanism of shipping around its mRNA or its genome. We don't exactly know. And interestingly, this protein ARC has been published 2 years ago in relation to herpes infections, which as well happens in neurons. And there, the authors could show that downregulation of ARC is actually a great way to reduce the spreading of herpes. So we published this paper in Science early May. Got a lot of attention, media attention. It was pretty exciting. There were 2 other studies at the same time, which found more or less the same as we did. One in Nature Medicine, a very elegant study by a lab in Hong Kong. And they had the nice addition that they also studied bat organoids from the horseshoe bat. They could use that as an infection model as well. And it could show that the horseshoe bat organoids from the intestine could very efficiently facilitate replication of the virus as well. And this is interesting because this could also mean that organoids in general could be used to study potential hosts of the virus. For example, pangolins or civets have all been suggested to be intermediate hosts for SARS-CoV-2 before the transfer to humans from the bat reservoir. And we think organoids could be a great way to study that potential hosts because some of these are endangered species. And just showing that also these authors noted that type 3 interferons are the ones that are most upregulated of an infection. So that's similar to our data. And the third story that came out also around the same time is this one in Science Immunology. And these authors as well could replicate the virus in different areas, even of the different organoids around, that were made from different areas of the human gastrointestinal tract. And as well, the virus could replicate in all these different organoids. And this particular study also showed that the virus is actually killed very efficiently by colonic fluids. So the fluids that exist in our human colon kills the virus. So that is important in terms of the spreading, that maybe the intestine is an important site of where symptoms can arise and where the virus can seed, but not so much where it can spread from. Because if it gets killed in the colon, we maybe should worry less about fecal spreading of the virus. And these are papers from the last, or this last paper is published, I think, a few weeks ago, expanding really to all organs. This is an iPS-derived organoid paper and some cell lines, where they really generated almost every tissue from iPS cells, the lab from Xubing Chen. And they could surprisingly show that the virus could also replicate in islet cells from the pancreas and hepatocytes from the liver. And this study is nice because many cells that they could culture here, for example, would never be possible from adult stem cell derived organoids, because the pancreatic islets, for example, are really cells that are formed during embryogenesis and don't have so much adult turnover anymore. So from an iPS derived system, these are relatively easy, or not easy, but easier to make compared to adult stem cell derived organoids. And to the right is the original paper from Joseph Penneger, already in April, where they used vascular, blood vessel, and kidney organoids as a model for viral infection. And they could show that soluble ACE2 could block the SARS-CoV-2 infection into these organoids. So we would like to take this research in our lab now to some more fundamental insights into the biology of the virus, by particularly focusing on genetic engineering of organoids. So we are now knocking out different genes which have been reported to be important for the viral infection or the response. So we're picking those from literature, or we are picking those from other coronaviruses, which have been described for other coronaviruses, but not necessarily for SARS-CoV-2. So we're making a bias selection here of genes to knock out. And secondly, we are making reporters of relevant genes so that we can follow those genes, we can follow the expression of those genes by fluorescent tags. Secondly, we would like to make co-cultures with immune cells. Obviously, the epithelial organoids don't have the immune component, but we can actively edit. The third line of research is maybe something we are not focusing on, but it is interesting to use adult stem cell-derived organoids for, we think, is as a drug screening platform. It's just a little bit difficult. What is your readout? Currently, it's difficult to measure high throughput, for example, the entry of the virus. But maybe when there are fluorescent reporters that it's possible. And fourth, we think that adult stem cell-derived organoids could be used to compare the efficiency of viral entry between patients. There is huge variability between patients on how susceptible people are to our SARS-CoV-2. And this is partly dictated by the immune system, but we think also the epithelium here could play an important role, and probably organoids could maybe play a role in this, in studying this. So just briefly on the immune cell co-cultures, this is a rapidly developing field. Two studies published 2 years ago in Cell, and one which is from our lab on bioarchive, where colorectal cancer organoids are co-cultured with T cells. And both studies could actually show that the T cells can actively kill the colorectal cancer organoids from patients. So making it a model to study the killing of cancer organoids. And we think we want to do this the same now with SARS-CoV-2 infected organoids and immune cells, and we can pick which immune cells to add based on the cytokines that we see that are upregulated in response to the infection. So the knockouts are really briefly introduced. We are making a bias selection of genes, so we're not doing a genome-wide screen, for example. It's very difficult in organoids. We're just picking a few dozen of genes, knocking those out, and they're mostly looking at how much does viral infection efficiency change, or by RNA sequencing, what is the response change? So we make loss of function mutations. So we either do that by using CRISPR-Cas9 to cut somewhere in the gene and create frame shift mutations. So this is a Cas9 that make double-strand breaks. And an alternative strategy that we are using is so-called base editors. These are Cas9 proteins that can actively edit individual bases in the genome. And we can basically try to edit certain codons that when you edit them, when you change the signal nucleotide, they will turn into a stop codon. So this is an alternative way to make knockouts. So there actually is one genome-wide screen just published on bioRxiv last week. This was done in Phero cells, so cells that really infect very well with any virus. Genome-wide, as I said, and they looked at basically knockout of genes that would render the cells less susceptible to die. So this was an apoptosis readout. So they would find genes that, when they are lost, will protect the cells against dying from infection of SARS-CoV-2. Of course, dying might not be the only effect of the virus. So yeah, that might be a downside, but the strength of the study was really that it is genome-wide, and that's something we're not reaching in organoids. And they really could find most of the genes that are expected. Surprisingly, not TMPRSS2, which maybe can be because Phero cells they're not the most physiological cell types, obviously, or because there is no effect on the cell death response. Already briefly introduced are the fluorescent knock-ins that we are making. So we can tag genes so they become fluorescent in organoids. Before, we were always doing this with homologous recombination. So we can use homologous recombination to introduce fluorescent proteins into the coding sequence. This is very laborious. You need to make the homology arms. Takes a lot of cloning and time. So now we are switching mostly towards a non-homologous end joining approach. And this was published 4 years ago in Nature Communications. It's called CRISPR-Paint. And what it basically does is you just introduce, using CRISPR-Cas9, a break in the gene, and then you provide the cells with a linear piece of DNA coding for the fluorescent protein, and it just can automatically insert in the gene, and then it will be a fusion. There are some tricks to make the insertion in frame, because you cannot exactly control the cutting side of the guide RNA of the Cas9. So there are some tricks to make it in frame, but in general, this is a strategy to make gene knock-ins to make fluorescent reporters. And our lab this year in Nature Cell Biology published this study showing that it also works in organoids, and it's a very quick method to tag any gene. So just briefly, we've subsequently, a few months ago, this got published in April, used this technique to label rare cells in the intestine. So we are studying the hormone-producing cells in the intestine, very rare cell types. Some of these cells are only 1 in 10,000 of the epithelium. And here we're using fluorescent reporters to mark these rare cell types so that out of a sudden they become accessible, because we can basically fish them out easily using FACS, for example. And this technique can also be applied serial times. So subsequent tagging can really make multicolor reporter organoids of many different genes. We have some organoids already with 3 different genes tagged, which really helps some of these this work. And also briefly, we have done this in snake venom gland organoids early this year, published in Cell, where we could tag in these snake venom gland organoids, we could tag toxins. The venom gland makes toxins, and we could use this technique to tag actual toxins. So for the SARS-CoV-2 work, we have now applied this for multiple genes which are relevant for SARS-CoV-2 infection. This is an example of TMPRSS2, already introduced by Anthony, protease that is important for the entry of the virus. Yeah, and this we think that these reporters can be useful to do drug screens that could, where we could look at molecules that modulate the expression of these genes. We can do lifestyle imaging upon infection. So for ACE2, for example, we have seen downregulation upon infection. We can use that using lifestyle imaging. And we want to use these reporters to compare the infection efficiency. So we can, for example, say, do ACE2 high cells, ACE2 medium cells, or low cells, which one get most efficiently affected. We can easily sort them. And this we can now do for multiple genes that are important for SARS-CoV-2 infection. And as I said, this will be interesting, we think, for ACE2, because we see many cells which almost have no visible ACE2 on the protein level, but still get infected. So what are the levels that are really important? And one other thing that we noticed is that in intestinal organoids, there is this very strong downregulation of the receptor, ACE2 receptor, upon infection. So here in red, the ACE2, and in white, the SARS-CoV-2 virus. And you can hopefully appreciate that the cells that are infected have way less ACE2 on the membrane. And we think this is because of endocytosis. Maybe this cell, you can here see this cell, which still has some granular ACE2 and got infected. So most likely this is endocytosis, but we hope maybe that we can visualize this and use reporters because these reporters are actually fusion. So in this case, ACE2 would really be fused to a fluorescent protein. So we can use lifestyle imaging to study this process. And with that, I would like to end, thanking mostly Hans, Maarten, and Jelte, with whom I'm doing the corona work, and Jens and Sherelle, who have really been helpful with the analysis and lab work in this project, and our collaborators in Rotterdam, the virologists, and transmission EM experts in Maastricht. And yes, with that, I would like to end.
Thank you very much, Hugh. A fantastic presentation. Really dramatic results with ACE2 and upon viral entry. So that's looking very exciting. Yes. So everyone, please type your questions in the GoToWebinar question box on the right side. So the Slido software is just for surveys. It cannot accept your questions, just to clarify that. We'll answer them all at the end. And now it's time for another slider question from, this time from you. This is one of Yu's questions. Describe what improvement you would like to see in your organoid system. Let's see what comes up.
Sure, not everyone works with organoids.
Oh, nothing. No improvement. It's
That's good to hear.
perfect cultures. I was expecting reproducibility. Wow, factor. Oh, reproducibility is coming up strong.
Drug screen makes sense.
What would you like to see? What would you like to see?
I would not use organoids. No, I can totally see the reproducibility point. That there are some patient variation. I can imagine that. I think for me personally, it's mostly the cell types. To make all the cell types in every organoid system is still the biggest challenge. To get all the different cell types. That's probably what I would, for most organoid systems, I think will be the biggest improvement.
Well, you mean using in core culture the different types of cells, like, or just single organoid?
Sorry?
So you refer to your presentation as the co-culture with immune cells. Do you mean this, or do you mean just organoids from different sources? Yeah.
Yeah, no, I mean really within the, for example, intestinal organoids. Now, intestine is the exception. We can really make all the cell types of the intestinal epithelium, but there are some tissues where we're still struggling to get all the different cell types.
Okay. All right. So this is fascinating. I like more yopness. Okay. Thank you. So
no.
now we have to move on. So now it's time for our final speaker for today, Dr. Jamie Van Essen. Jamie completed her PhD in biological chemistry in Aaron Goldstrom's lab, University of Michigan, Ann Arbor. She studied the mechanisms of gene regulation by the PUF family of RNA binding proteins. Jamie then went on to a postdoc in cancer biology in Stockton's laboratory, Minnesota. During this time, Jamie focused on alternative polyadenylation of the androgen receptor in prostate cancer, where she developed a morpholino therapeutic targeting the active androgen receptor. Jamie is recently joined by Bio-Techne in the stem cell and gene therapy group, where she has developed or accelerate human NK cell growth medium. For today's presentations, Jamie will be focusing on methods and reagents for profiling innate and adaptive immune cells in COVID-19, exploring more in vitro approaches which mimic the cytokine release syndrome and its impact on T and NK cell function. Take it away, Jamie.
Hey everyone, good morning, good afternoon, good evening, wherever you are. Thank you, Yaz, for the kind introduction and for the invitation to speak today. I'm really pleased to be here, and I'm looking forward to your questions later on. So, like many of you, I was not expecting to be here today in the midst of a global pandemic. And I really hope that we can share our questions and our expertise and our visions for the future as we really ramp up during this time our research to understand COVID-19. And here at Bio-Techne, we're working really hard to develop reagents that you will find useful as you move forward with these projects and the many that may follow. All right. So like I said, here at Bio-Techne, we have a lot of resources to get you moving in terms of immunity-related research. So we offer streamlined and scalable immune cell expansion reagents, and those include our clouds microspheres for expansion of immune cells, our accelerate media products, so that's our NK cell, which I've developed, our T cell media and a B cell media, and then as well as supplements and cytokines and proteins. And Tony did a really nice job of really taking you through a lot of the available proteins out there. In addition to just growing your immune cells, we offer a lot of solutions for cytokine profiling using ELISA-based methods. So our Luminex multiplex platform with our conjugated beads, the ELA system and Quantikine, which are just your basic ELISA format. And if you're looking at more Western blotting, we have Wes and Jess and some of the technologies over at Protein Simple, which will accelerate the speed at which you can perform a lot of these assays. In addition, once you've grown up your immune cells and treated them with whatever you plan on treating them with, or if you've infected them with virus, we've got a lot of reagents, particularly in the realm of flow cytometry antibodies for profiling those immune cells, whether you're looking at surface markers or intracellular markers. In any of your different immune populations, we likely have an antibody that will work well for you. And if we don't, talk to us. Additionally, we are in the midst of developing new protocols for cytotoxicity-based assays. So if you're looking at the impact, so I'll talk a little bit later about the impact of different cytokines on cytotoxicity. We have protocols that are working really well to assess changes in your cytotoxicity in different immune cells. And now, if you really are looking to make changes to your primary cell lines, the gene editing wizards over at Beamogen have excellent non-viral based gene editing tools for making CARs, for making TCR expressing Tregs, you name it, they can edit it. And so if that's something that you are in need of a reagent for or a custom, if you need to make a change in your favorite cell, those are the guys to go to. Of course, you still need to get to the lab, and I'm hoping that many of you are able to get back to the lab soon. So for those of us who are maybe not immunologists by training, I thought I would give a brief overview. So our immune system serves to protect us from disease and other damage, and to successfully make a distinction between self and non-self in these instances. And so this system uses anatomical barriers like our skin to avoid infection and other insults. The humoral system enacts antibodies and proteins and antimicrobial peptides, which are secreted in our extracellular fluids in order to destroy foreign invaders. Our innate immune system is an ancient and very highly tuned cell-mediated system of cells, which are nonspecific, and those use germline encoded receptors to identify stressed cells. So a virally infected cell, a tumorigenic cell. And this includes cells like natural killer cells, which will really be the focus of my talk today, macrophages, and neutrophils. And our adaptive immune system is a lot slower, but more specific. And this has memory of past infections and insults to the immune system. And this includes B cells and T cells. And so, like I said, we'll focus on natural killer cells today, as well as T cells for COVID-19 related work. So let's talk about NK cells. So natural killer cells recognize stressed cells. So this can be due to tumor transformation or DNA damage, microbial infection, and many other issues. So these cells are activated by antibodies or interferons or cytokines, and sometimes by other cell types. And this really gives them wide-ranging sensory function in the body. And once they're activated, they induce apoptosis. And they do this by releasing granules that contain enzymes that can make pores in infected cells or tumorigenic cells. Then they break down cellular proteins, and then they kill these cells via release of pro-inflammatory cytokines. They can also recruit other cell types. And they can also act by antibody-dependent cell-mediated cytotoxicity, and that's via the CD16 surface marker. So we'll do a quick run through of kind of a general protocol that we would use in the lab to expand NK cells from peripheral blood. So if you're looking at using a primary cell, you can use peripheral blood derived NK cells, but they're fairly low in their in the abundance in the peripheral blood. So in your PBMC culture, you may start with only about 10% of that culture being NK cells. So if you need to grow up a large quantity of NK cells, we have a really nice system for growing up these cells and activating them and expanding them in culture so that you can use them for experiments. And for those of you who are interested in a more clinical pipeline, for those purposes as well. And so this is just my general protocol, and this is what I typically use for an expansion type protocol. And this is using both Accelerate NK expansion media and our NK clouds microspheres. And those are conjugated with antibodies that activate the NKs, and then we add additional cytokine. And we have kind of our favorite cocktail of NK cytokines, which are IL-2, IL-12, 18, and 21. And these concentrations we have found to be kind of the Goldilocks of concentrations in our hands. And obviously this protocol is flexible for you. And so the idea here is to isolate our PBMCs. We typically just, isolate the buffy coats by a centrifugation with like a sepmate tube. And then on our first day, you can use either fresh or frozen PBMCs. And as long as you seed around about 200,000 NKs, so you can confirm that by flow cytometry, 200,000 NKs per mil seeding density, that seems to work pretty well in our hands. And then add your clouds, add your cytokines and your media, and let it sit for a while. And you add and change media every 2 to 3 days. We typically supplement and feed with a double concentration of cytokine, just assuming that a lot of those cytokines get used up. And then if you need to, you can split as needed. And at day 10 or day 14, in serum-free conditions, you should have a pretty robust expansion of NKs. And so all that's nice, but let's show you the data. So this is an expansion of NKs using Accelerate serum-free media, our NK clouds, and our GMP cytokines. And this is PBMCs from 3 different donors. And so as you can see, we've stained our NK cells with CD56, which is a known surface marker, probably the most reliable surface marker of NK cells, and CD3. So NK cells are CD3 negative. CD3 is really the universal marker of a T cell. And so we look at the CD3 negative, CD56 positive population when we're trying to identify expanded NK cells. And so you can see here that we're seeing purity above 75% for 3 donors after 14 days. And we'll see a range. Some donors are better than others, but this is pretty representative of what you might expect to see. And then in terms of fold expansion, we just look at the total number of NKs at the end of the experiment, and we calculate that based on cell counts, so live cell counts, and then the percentage of CD56 positive, CD3 negative cells. And so then we calculate that based on our seeding density. And so in our Accelerate media, we typically see expansion above 50-fold after 14 days, and this is serum-free and feeder-free, feeder cell-free conditions. And some donors we see, I think I had a super donor one time that was like 2,500-fold expansion, but I think that was a unicorn because this is more along the lines of what we would expect to see. So my video is not working. There we go. All right. So when you're expanding your cells, you want to make sure that you have a functional population of natural killers. And so we can measure cytokine release using multiplex cytokine profiling assays, which I'll cover in a little bit. We can also use a dye-loaded target cell that leaks its dyes upon attack by a natural killer cell. And in our lab, we typically use the K562 myelogenous leukemia line. Those are well documented and have a known susceptibility to NK cytotoxicity. And so I'll show the results of a dye leakage experiment shortly. But here we used some really cool new technology from the folks at PHI. So this is a phase holographic imaging system. So this group is in Sweden, and this is imaging K562 is under kind of assault by natural killer cells. And so you can see, run that again. It's not letting me run it again. There we go. You can see this central guy, that's our K562. And all of these NK cells are kind of swarming around it, and that central cell has really just been obliterated. And we see this time and time again. And I just thought that this is a cool way to represent the process of NK cells taking out a cancer cell. They really do. It's fascinating to look at this under a microscope. All right, so in COVID, infection by this SARS-CoV-2 and the resulting COVID-19 disease results in an activation of a huge number of immune cells, and that includes monocytes and dendritic cells and macrophages. And one of the biggest problems that we're seeing in the clinic right now is this cytokine release syndrome. You're hearing about it all over the news. You're hearing about it in every scientific journal out there. And these cells are releasing massive quantities of cytokines in response to viral infection. And so typically IL-6 is a big driver of this, as well as IL-8, interferons, TNF-alpha. And this upregulation of IL-6 is initiating signaling and amplifying cascades that result in downstream trans signaling in a lot of other immune cell types, as well as other cell types, so like endothelial cells. And so this results in this increased, probably exponentially increased systemic inflammation and this severe pathophysiology that we're seeing in patients who have this disease. And IL-6 is known to play a role in this acute phase reaction, as well as in inflammation. And so we were particularly interested in the IL-6 signaling methods. And so some serological features of severe COVID-19 infection: really high IL-2 receptor, IL-6, IL-8, IL-10, TNF, high procalcitonin, serum ferritin, and high C-reactive proteins. These are all kind of markers of severe inflammation in a patient. And critical patients with COVID-19 are lymphopenic, which means that they have really low levels of lymphocytes. And so they have high neutrophils and a low level of NK cells, T cells, monocytes, basophils, and eosinophils. And there's some evidence out there currently indicating that a neutrophil to lymphocyte count, a high ratio of neutrophils to lymphocytes, is possibly prognostic for severity of disease. And that's something to pay attention to going forward. And so today we're going to focus on IL-6. And the reason for that is that there is a huge number of publications currently and in past SARS research, MERS research, that suggests that this high IL-6 serum level is really promoting immune dysfunction and dysregulation in patients with COVID-19. And so here is just, I took screenshots of a handful of papers. There are many more. But in several studies, we see that the level of IL-6 in the serum is predictive of respiratory failure and critical illness in COVID-19 patients, and that this JCI paper, which you can see down here, has a really interesting, it's kind of a small patient cohort, but they look at impaired immune cell cytotoxicity, and they show in their paper that this is dependent on IL-6. So we asked, what is the impact of high IL-6 on NK and T cell function? In our lab, we are fortunate because we can grow NK cells, we can grow T cells and expand them in order to do all kinds of studies. And we have a lot of different cytokines that we can try to mimic this cytokine response syndrome. And so we thought it would be an interesting study to do more of an in vitro approach to look at the impact directly on NK cells and T cells. And so here is our expansion protocol. So you'll notice that this is a little bit different than my first workflow. And so in this particular experiment, I looked at NK cells from the peripheral blood from fresh PBMCs from 3 donors. And I seeded at the normal concentration, and I let those cells grow for about 7 days so that I increased my pool of NK cells in my PBMC population. And then on day 6, I added my test compounds, I let that sit for 3 days, and then I harvested the cells by looking at flow cytometry markers, so for surface markers, and then I also pulled the supernatants in order to do a Luminex panel on these to see if there were really any differences between the conditions. And so the test compounds that I used 50 nanograms per mil IL-6. I also used a neutralizing antibody for IL-6, which is called sarilumab. You may have heard of that. And also a gallialectone, which is a STAT3 inhibitor. So IL-6 upregulation is thought to occur through this STAT3 pathway. And so I thought that that might be another, so in addition to a neutralizing antibody, that would also kind of rescue a phenotype if we were going to see one with IL-6 treatment. So in order to look at the supernatants, I used Luminex technology, which is a really nice multiplex system for ELISA-based detection. And so what's really important about this is that you're not restricted to certain panels or a bead format. You can get up to 100-plex or 50-plex depending on the bead identity. These are user-defined and pre-mixed, and you can use, we don't just have human analytes available, we also have rat and mouse. And this is just a 2-laser detection system. So one laser distinguishes the bead and the analyte, and the other reads the biotin streptavidin signal. And you can simultaneously look at either 50 or 100 different analytes at a time and get down to picogram detection levels. So in my experiment with a pure population of NK cells and 3-day treatment with IL-6 at 50 nanograms per mil, I was really surprised to see that very little changed in the supernatant. So what these cells were secreting into their cell culture media was really not very different depending on whether or not there was IL-6 present, a neutralizing antibody, or this STAT3 inhibitor. And so I decided to change up the way that I was doing my experiment. I thought maybe there is an impact on NK function, but I have to start treatment early on. And so the next experiment that I did was I did the same seeding procedure. So this time I had frozen PBMCs from the same donors I had tried in the previous experiment. And instead of waiting for a week to let them grow up and then treat them, I treated them with different concentrations of IL-6, as well as the neutralizing antibody for IL-6, the sarilumab. And this time I left out the STAT3 inhibitor because I really wanted to see if we could tease out an effect of IL-6 on NK function. And while I don't have the Luminex data for that, it just sort of is rolling out, and these have not been processed yet because these experiments take two weeks minimally. What I did find was, in terms of looking at purity of my NK or my PBMC population, so how many NKs do I have in my culture dish at the end of a 2-week growth experiment, I didn't really see a big impact of IL-6 on identity of NKs, purity of NKs. And so that's looking at the CD56, CD3 percentages in my populations. And then I also looked at growth, so overall expansion at the end of 2 weeks. And I have, I have 3 PBMC donors here, and it looks like there may be a slight decrease in fold expansion at the end of 2 weeks. It does not yet reach statistical significance. I think that in a larger experiment with more donors, we may be able to get a little more power behind this and tease out this effect. But I don't see a huge difference on NK expansion from PBMCs when they're grown in continuous IL-6, which would really mimic that cytokine release phenotype. But when I look at these cells by cytotoxicity assay, so this is K562 loaded with calcine AM. So when you load up the cells, they shine brightly green, and then you add your natural killer cell population. So this is done at day 14, at the end of my expansion. I have a nice and very pure population of NK cells. I add those at different effector to target cell ratios. So this particular experiment, I did a 5-to-1 NK to K562 cell ratio. And then I just continued to, I let these sit for about 4 hours at 37 degrees. So as the NK cells kill the target cells, so these K562 cells, they start to leak the green dye out of their, as they die, basically, into the supernatant. And so at the end of this prescribed time period, you can go between about 2 and 4 hours and you'll see nice killing. You spin your cells down and then collect the supernatant and look at that by fluorescence absorption, or sorry, fluorescence on a plate reader. And so what I saw here is that treatment with IL-6 reduced moderately, at least, NK cell cytotoxicity. And this is the combination of 3 donors and 3 technical replicates. Interestingly enough, my neutralizing antibody didn't really have an impact on rescuing this effect by IL-6. It would be worth trying other IL-6 neutralizing antibodies, but in this case, we didn't really see an impact. But it is comforting to see that IL-6 can reduce the cytotoxicity of NK cells. And so then we switched gears and looked at T cells. So here's another workflow. The general idea here is to use the accelerate T cell media and T cell clouds. And so those are conjugated with CD3 and CD28, as well as T cell specific activating cytokines, IL-2, IL-7, and IL-15. And then at days 5, 7, and 9, we assess by CD25 staining and CD69 staining, as well as counts for T cell expansion and purity, just like I described for the NK cells. And so typically in these experiments, you use about 50 microliters of clouds for every 1 million starting cells. So it's slightly different than the NK protocol. And so what we saw in these experiments was that in the presence of IL-6, we observed an augmentation of T cell proliferation in the presence of IL-2, as well as in the absence of IL-2. And this was normalized. So on the left panel, you have the presence of IL-2, so you see about a 36% increase. And compared with IL-2 alone, we see an increase in T cell expansion. And so that was really interesting. We next wondered if IL-6 affected T cell function. But here in this is the same cytokine panel that we used for our NK cells. So at certain time points, you take the soup and send them, you do your Luminex multiplex assay here. And so we didn't see any major changes in the phenotyping of these cells. So for example, the percentage of CD4s, CD8s didn't really change. So we looked at the cytokine secretion protocol. And so again, this is using our Luminex 2-laser detection system. And we looked at up to 50 cytokines at a time. And on the right, you see just a few cytokines in 15 different conditions in this experiment. So this is a huge amount of data that we've generated. And so we really pulled out what looked interesting to us. And so here's kind of a zoom in on a couple of pieces of data from the T cell experiment. And so what we saw was that elevated IL-6 might affect T cell function and cytotoxicity. Normalizing for the cell number, one challenge that we came across is that every experiment, at the end of the experiment, you have a very different number of cells. If you have a cytokine added that's affecting proliferation, at the end of your experiment, in your cytokine, you've got a different number of cells producing cytokines. And so we thought we need to really normalize this to the total number of cells that are in there. And what we saw was that normalizing for cell numbers, the cytotoxic cytokine interferon gamma decreases, as well as granzyme B. Fractalkine increases, which is typically increased in inflammation. It's been noted in pulmonary inflammation, so it might be related to COVID-19. The FLT3 ligand is related to expansion of blood cells. It also promotes the differentiation of multiple hematopoietic cell lineages, like monocytes, dendritic cells, and early B cell lineage. And so that could be something that's really reflective of this increased T cell expansion. So to summarize, the impact of IL-6 in ex vivo or in vitro purified and expanded NK and T cells, the impact on NK cells here is that it decreases NK cell proliferation just slightly and moderately reduces NK cell toxicity against K562 cells. And on T cells, we see an increase in T cell proliferation, but per cell, IL-6 treated T cells make more fractalkine, which is an inflammatory marker. And per cell, IL-6 treated T cells make less interferon gamma and granzyme B, suggesting a reduced toxicity. And so here, we really focused on T cells and NKs, but the effects in vivo are much more complex, and they likely involve a lot of other immune cells, such as macrophages and B cells. And the ex vivo cultivation and experiments that we have possible with these workflows allows an understanding of how individual immune cell types respond to aspects of COVID-19 infection, like IL-6. And so I hope that we've illustrated, you can use a lot of these reagents to do some tests on independent populations of immune cells that hopefully will help us get an idea of what's really going on here. And so here's just a workflow of all the reagents and tests we have available for SARS-CoV research in the area of immunity. On the left, I've got the NK expansion and activation reagents, so our NK clouds, our accelerate serum-free media, T cell activation and expansion reagents, so again, that accelerate T cell and T cell clouds. Like Tony mentioned, we have a lot of GMP proteins available. We have PRODOTs available if that's something you're interested in. They're lyophilized proteins, typically geared toward more of a clinical workflow. For characterization, we have a huge library of flow cytometry antibodies, Luminex, multiplex detection of cytokines and chemokines. We also have ELA, which does similar cytokine profiling. And then for further research, pathway-based, signal transduction-based viral research, we have a repurposed drug library for SARS-CoV-2. We also have an in situ hybridization probe for SARS-CoV-2, and that's offered through ACD. And then any sort of protein profiling you want to do and save yourself the time on a Western blot, we've got this simple Western line of Western blotting products like Wes and Jess. And then if you're interested in scaling up to a immune cell therapy workflow, so there are some studies ongoing in COVID-19, really severe COVID-19 infection, looking at the possibility of using NK cells, activated ex vivo, and then treating patients with those NK cells to try to dampen the viral response and kill off virally infected cells. I do know of at least 1 or 2 ongoing clinical trials that are using that. We're trying to cover all of our steps in terms of scaling up this as a cell therapy workflow, so you can get in touch with us if that's something that you're interested in as well. And with that, I thank you for your attention. It's really fun to be here. And I'm looking forward to all of your questions.
Hi, thank you so much, Jamie. That was great. I mean, I think for me, I've heard so much around the health implications of COVID. It's great to see some fresh data from the lab, and it's nice to see the technologies that's already been developed and we've developed, and the technologies being developed. So I always think it's a treat to hear about the people, our scientists that are developing the technology as we go around. So thanks so much. Team worked hard to get us fresh data, so it's really, really appreciated. Okay, everyone, so please type any questions that you may have in the questions box for GoToWebinar. It's on the right side, and we will all ask them all in shortly. And then, so now, just to keep it fun, some more Slido questions. Next set, please. Yeah, so what's your main interest in COVID-19 research? What are you researching? What do you need from us? So, so you've heard all about what we're doing, but it'd be good to see what you need from us and how we can help. So it's a free word association. Okay, so diagnostic. Okay, cytokine storm. Okay, cytokine storm is coming up quite strong. Right. So when the letters, the biggest letter is the one that most people are typing. So the size indicates level of interest. Okay. So some people are working on the immune response, some diagnostics coming through, some patient interventions, some people are looking for new targets. Okay. All right. Okay. Ooh, we give it a bit more. Okay. Okay, so a lot is coming around therapeutics, cytokine storm, immunity. Okay, we have to move on. Ooh, people are starting to type now. Just give it a bit more. Okay, so everyone, time for the next question, please. Yeah, so it sort of follows on from the last, but if you had your wish of any COVID-19 research reagent or therapy, what would you wish for us to develop, or what would you wish for generally? So, immunomodulation, faster, rapid, antivirals, okay. S2 subunit proteins, okay. There's a little bit of a delay in, I think, in the software to see all the responses, but it's all coming up now. Okay, ACE2 receptor inhibitor, right? Blocking proteins, more antibodies. All right, tools to immunomodulate seems to be winning with a note, fast response, I guess. All right, everyone, thank you so much for participating in the surveys, and I can also reveal to you now that our next upcoming symposium is on targeted protein degradation, TPD. So if you're unfamiliar with this, TPD is a new fast-growing field. It's a technology that harnesses the modalities of small molecules. I guess the most well-known is PROTAC degraders. So these molecules, they achieve a selective knockdown of target proteins within cells. So they're sort of a new, more effective means of creating knockdowns. So, and these offer exciting potentials because they are expanding the available drug targets in the proteome, and they're more effective. So, for those of you who've been typing drug screening, drug targets, therapeutics, this might be an event that might be of interest. So there is a link in the abstract booklet if you want to find out more and if you want to register. There's also a live poll coming up. So if you're interested, please just say yes, and you will get the registration link sent to you direct. All right. So we give just a little bit of time for people to be able to vote. All right. Okay, so we have roughly 10 minutes for the question and answer. So I think we'll move on to the panel. So, all right. So we'll start everyone with some general questions. So I think there's a lot coming up with, obviously there's a lot of current interest in the therapies. I know, Anthony, you started with discussing all the different types of proteins and the targets, the proteases as targets. If I'd like to ask each of you, where do you see the most therapeutic, the most promising therapeutic avenue right now? Is it vaccines? Is it more for cell-based therapy, or is it blocking various proteins? So if we start with Anthony, please.
Sure. I think that sure. As I alluded to in my presentation, I think that it's clear that the pharmaceutical industry is getting very good at making therapeutic antibodies. You know, if you look at the TV and all the new drugs that come out on the market, there's so many monoclonal therapies. So I do believe therapeutic anti-spike or ACE2 antibodies will be part of the treatment regimen. I think another area that's really going to be important is the protease biology, as I alluded to. Classic small molecule steric hindrance type drugs will be designed, we would think, for the papain-like proteases as well as the 3CL. In addition, I believe that although PROTACs or targeted protein degraders are also a very new drug modality, that there's going to be effort in the pharmaceutical industry for targeting those same proteases with PROTAC reagents. So I think it would be blocking antibodies, small molecules, and even PROTACs may come into play here. The vaccine, who knows how well a vaccine will work? I know we have many companies working on that, and it's a race, but the efficacy of that approach is still unknown. So that's my opinion on those questions.
Thank you, Anthony. What about yourself, you?
I must say it's not completely my area. I had pretty good hopes on vaccines, but I got a little bit less enthusiastic now after hearing Anthony. Yeah, I think what Anthony, I'm totally with Anthony. Those targeted therapies look very, very promising. Yeah, from our side, I hope that we can identify some new therapeutic targets with knockout screens that could help. But I find it difficult to make any predictions there.
Thank you. It's the $1 billion question.
Yeah, probably a little bit more than that.
We can't have a symposium on COVID and not ask it. What do you think, Jamie?
I agree that targeted therapies, monoclonal or these PROTACs, are going to be more rapid to get into the clinic. I'm with Tony and Yup, like vaccine development is slow. And as quickly as people are moving, we just don't know. It's a high risk, potentially high reward endpoint. But in the meantime, we have a lot of people who are sick and need to be treated. And so these monoclonals that, if they prove efficacious early on, I think that they'll be really useful. And, hopefully when we get a vaccine, great. But that's a long way off. I think in terms of immune cell, I know I'm here to represent the immune system.
Yeah, NK cells.
I love NK cells, but you see in these patients who are so severely ill, the immune system turns on itself in this cytokine storm, and until we understand the biology a little more fully, I know that there's a recent study that's showing that dexamethasone is having a good effect on severely ill COVID patients because it's dampening that response. But if you treat them too early with that, it could kill them, right? So that I think we really need to understand that biology before we put too much effort into developing, an immune cell therapy for COVID, because we just don't know what that impact will be. So I think the real to represent the work that I'm doing, we really need to understand the biology of what's happening with these immune cells and really understand what's going on there with viral infection. But these targeted therapies, I think, will be a good solution, especially if the vaccines aren't terribly effective, which we just don't know.
Yeah, I'm still sort of having one bet on NK cell-based therapy a little.
I mean, it would be awesome. And if you could treat a patient early, If you could profile their NK cells and see how cytotoxic they are, where are they in this process of immune cell dampening? Because there's some evidence that shows that that's happening too. So if there's early inactivation of those cells, if you could then promote an NK activity or highly active NK therapy you could get from a donor population, or, I think it's going to be timing so that you don't make people too sick or not have any.
All right, so we have some specific questions for our various presenters. So I think I'll just select a few. Anthony, for you, we have a question on glycosylation of the key protein. So one question is, since glycosylation appears not to, on the spike proteins, appears not to affect the binding to ACE2, what do we think it represents, or what kind of function would it be performing?
Yeah, I think
question.
Sure. I think it comes more into play in the context of making relevant blocking antibodies if you're going to use these proteins as antigens. In addition, I think it's more important if you're using, let's say, a spike protein in an antigen down ELISA serological diagnostic test or a lateral flow test, and that remains to be tested. We have a rich history of glycobiology reagents, and we're kind of playing in this realm to design new, faster ways to glycoprofile proteins. This work is still in progress. We want to make these reagents available to our customers, but we kind of get sick of having to do mass spec on every protein that we purify because it's very low throughput and cost intensive. So I guess the jury remains out on that. But we're also using this sort of approach for our monoclonal antibodies. If you look at the high mannose N-glycans that we can probe for, that's the unwanted, more immature N-glycans on therapeutic antibodies. We think that this approach could allow us to test in a more efficient way to see if we have more mature N-glycans or less mature high mannose glycans on our antibodies that we're developing as well. So in this context, not sure yet. It's a new tool, and we have more analysis yet to do to understand how these glycopatterns are affecting the protein binding as well as antibody generation against these proteins.
Okay, so one more question, but in the interest of time, if it's possible to give a quick answer, Anthony, it would be great. Otherwise,
I'll try.
Or we all get told off by Luke for going over time. So the second question is, what is the difference in ACE2 binding affinities of different glycosylated spike proteins and the bacterial non-glycosylated spike protein?
That's a good question. We have yet to make E. coli-derived versions of the spike proteins. They're heavily glycosylated, and we wanted to go right to mammalian systems to make them. If you look at the full-length spike proteins, there's over 20 N-glycosylations on it. So we thought it was important to go right into the HEC, CHO, and even insect systems to make that. We haven't made an E. coli-derived version yet, so I don't know the answer to that, to be honest. Be an interesting experiment to try, though.
Everyone watch this space, you will know once we work it out. And then some questions directly for yourself, Yup. Okay. So there's a question about just specifically about lung organoids, where you've tried developing and establishing lung organoids, or the lab has, and what's been your experience on that?
Yeah, I didn't show anything of that today. So our lab works with lung organoids. They're more of the bronchus and bronchiole rather than the alveoli. So we don't have good alveolar cultures, which would be also very important in relation to COVID-19. So the infection is not as efficient yet as we have in the intestine, but we're working on that. So that's also ongoing work that I didn't show today, but certainly we're working with lung organoids in relation to corona infections as well. The genetic engineering is there also a little bit more tricky. So for the intestinal organoids, the intestine is a great system to make any mutations in a very physiological system of ACE2 or any other gene. It's easy in the intestine. It's not simple in any lung organoid system.
Okay, so we have one more question that we can ask. Is there a second possible entrance mechanism for the virus? Particularly interesting that expanding organoids have such a high virus expansion.
Yeah, I think that's a very good question. So we were a bit surprised to see that expanding cells with low ACE2 have such efficient infection. So it could be that there's just not so much ACE2 needed, and ACE2 is still the only and primary entry receptor. That could very well be. So we'll see that how cells perform when they don't have any ACE2. I think it could very well be their second entry receptors. The corona SARS-CoV-2 has been shown to bind to DPP4, which is the entry receptor for MERS. So it's not unlikely that there are alternative pathways to shuttle in the virus, and we hope to find these with knockout screens in organoids.
I mean, there's a lot of questions that we can ask. I thought that your the intelligence on the ARC protein is very interesting, but we leave this for now. So Jamie, thank you, Joep, thank you, Anthony. So we asked a few quick questions before we get cut off. Jamie, we have one question: could the local secretion of cytokines from the infected epithelium affect NK killing? And then do you reckon these are type 1 and type 3 interferons?
That is a really good question. I suspect that it could. I think it depends on what's present and what's being secreted, but I think that there's a number of ways that these things are getting shut off or hyperactivated and dysregulated, I guess you could say. And so I do think that is a possibility. Whether they're type 1 or type 3, I don't know. And that may be my naivete, but I'm not actually sure. And I'm not sure, I haven't seen a whole lot in the literature to date that really shows what's happening in the epithelium. My suspicion is the NKs are having a much broader impact on the epithelial tissue versus the other way, but I'm not sure.
So we asked one more question, everyone, and then we have to email the answers because we're fast running out of time. So Jamie, why do you study? Why pick the cytokine profile in NK cells if we have data that cytokine storm was released by macrophages, monocytes?
That's a great question. We wanted to understand the specific impact of cytokines being released by our NK cells. We actually really wanted to see what happened to interferon gamma and granzyme, things like perforin. And we're waiting to get that data back because the cells were so activated that they're beyond the detection limit, and we just keep doing dilutions and dilutions and dilutions, and they're still too far beyond what we can detect. So we wanted to understand, regardless of how this cytokine storm comes to be, what is the impact of those cytokines on signaling by our NK cells? And so we had a really unique opportunity with an NK population, and some data I didn't show was just from a pure population of NKs that I started, because I only have one donor, so I want to show a lot of data with just one donor. That's not, I want to see more. Do these cytokines that would be released by other cells have an impact on what NK cells are secreting? Because that in turn will change the way that they're either killing or not killing target cells, or maybe they're inappropriately signaling. And so that's why we did it. And we assumed that we'd have a much purer population to look at, and assuming that most of what we're seeing there is coming from these NK cells.
Okay, thank you, Jamie. Thanks to the panel for answering the questions. There is a few more, but we get them answered and we email it to everyone. And just want to say very briefly, for the people who've written asking for attendance certificates, if you need that, please put it in the question box. We can take care of that. We have certificates, attendance certificates we can give you And then, so, okay. So everyone, thank you. So I'd like to, first of all, take this opportunity to thank the entire Bio-Techne team who worked so hard and enthusiasm to realize the symposium. Huge special thanks to our guest speakers who've shared their time and research. And I want to make a special mention of our lab, scientists in our lab who are working all out to make the COVID, SARS-CoV viral proteins and relating COVID proteins and reagents. So it's not easy to work in the lockdown. So it's like a big thank you to them, and thank you to all of you for attending and staying with us for the duration on a very hot day in Europe, at least. So
Yeah.
we found that,
Yes.
we really hope that you found these sessions of interest, and we look forward to you joining us, hopefully for future events. Don't forget about the golden tickets, please. Don't forget about, and we wish everyone a great, safe day ahead. Thank you so much.
Bye now.
Thanks.
Bye.
Bye.